Hello, and welcome to my physiological mechanism teaching video. Today I'll be going over a synapse. To start off here, we're going to go over an electrical synapse. And I've drawn here a diagram of two cells. We have the plasma membranes. We have one on the top and then one on the bottom. And they're connected with these gap junctions here in the purple. And what these gap junctions do is they allow ions, molecules, and therefore action potentials to move throughout the cells that they connect. And so let's say for cardiac muscle, these are in high numbers in cardiac muscle. And so what this does is it allows that muscle to contract as a unit compared to separate contractions. So that's very important for the heart and for the cardiac muscle. So these gap junctions are vital to the heart working properly. Moving on to chemical synapse. Here we have a presynaptic neuron in the red and a postsynaptic neuron in the brown here in the bottom. And we have action potentials moving down the axon of the presynaptic neuron, and they're in the green arrows, and they're moving into the terminal buton, this lower portion of the presynaptic neuron. And once they reach the terminal buton, they allow calcium channels, which are the teal color surrounding the terminal buton. They allow that calcium to be released from those channels, and this calcium then binds to a sensor protein. And once this combination happens, it allows for a fusion and then the release of a synaptic vesicle. So going back to a synaptic vesicle, we have they're in the burgundy or maroon color here, and the neurotransmitters or chemicals are within the synaptic vesicles. And before the action potentials even reach the terminal buton, these synaptic vesicles are docked to the synaptic cleft with a snare complex. And so we have a snare complex, then we have a synaptic vesicle attached to it, and then those two are then attached to the synaptic cleft. And so the first step is docking of the synaptic vesicle, and then with the calcium and sensor protein combination, it allows that vesicle to then fuse to the synaptic cleft, and then it allows the release of that synaptic vesicle and the neurotransmitters within it to be released. So now we have neurotransmitters released into the space separating the postsynaptic neuron and the presynaptic neuron. And so now these neurotransmitters are then referred to as a ligand, and these ligands then attach to receptor proteins on the postsynaptic neuron. And once the specific neurotransmitter attaches to a receptor protein, it's very important to remember that the receptor protein is very, very specific. And so once that specific ligand attaches to receptor protein, it then allows chemically regulated gates to open. So based off of the neurotransmitter that was received, or the ligand that was received, it'll either release a positive ion or a negative ion into the postsynaptic neuron. And so let's say a positive ion was released into the postsynaptic neuron. That would be an excitatory postsynaptic potential, which would allow for depolarization and be closer to threshold. If a negative ion was released into the postsynaptic neuron, it would be an inhibitory postsynaptic potential, therefore hyperpolarizing the neuron and preventing it from getting closer to threshold. It would actually get further from threshold. So let's say that a positive ion was released into the postsynaptic neuron. Now we have an excitatory postsynaptic potential, and this allows for depolarization of the neuron, and it spreads throughout the dendrites, and then it goes to the axon initial segment, and where it then allows for sodium and potassium to be released into the axon, which creates that action potential. And so this action potential is then transferred throughout the axon and sent down through the rest of it. So that is a chemical synapse. And that is it. Thank you.